In this lecture, we're going to look at three virus families that have multipartite or multiple segmented negative sense RNA genomes. The first family we're going to talk about would be the Bunyaviridae. Members of the family Bunyaviridae infect plants, insects, and vertebrates. Bunyaviruses are enveloped, segmented, negative strand RNA viruses. The virion structure is pretty simple. Structural proteins include a nucleocapsid or N protein and two transmembrane proteins, GN and GC. The L polymerase, or the RNA-dependent RNA polymerase, is also present in the virion. Bunyaviruses have a replication cycle similar to that of other negative strand RNA viruses. However, some bunyaviruses have ambisense genome segments. We'll talk more about that in a bit. Bunyaviruses can cause severe disease in humans and substantial economic loss through infections of domestic livestock and plants. Most bunyaviruses are transmitted by insect vectors. Notable exceptions are members of the genus Hantavirus, which have rodent hosts and are occasionally transmitted to humans via rodent feces and urine. But in this illustration, you can see that there are three RNPs inside the enveloped virus, and down below you can see the three genome segments in terms of their order and size, so small, medium, and large size segments. When we look at the infection cycle, the first step involves an interaction between cell surface receptors and viral attachment proteins, GC and or GN. The functions of each may vary among the genera. Both may be needed in some cases. Or, for instance, it could be that one is used for vertebrate cells and one for mosquitoes. So, for instance, lacrosse virus is transmitted by mosquitoes, but it infects vertebrates. So it's believed that GC is the glycoprotein anti-receptor used for vertebrates, and GN is the one that's used for mosquitoes. Host cell receptors have not been identified for most viruses in the family. Entry and uncoding is by exocytosis of virions and fusion of viral membranes with endosomal membranes. So, the sort of thing that we've talked about before. Acidification of the endosomes promotes a conformational change in GC that facilitates fusion of the viral and cell membranes. Transcription starts by viral RNA-dependent RNA polymerase, or the L protein, binding to a promoter on each encapsulated segment, and is terminated by a strong hairpin sequence at the end of each gene. The mRNAs are capped by L protein during synthesis using a cap snatching process. That is, they actually cleave the caps off of cellular messenger RNA and use that. However, the mRNAs are not polyadenylated. The caps are necessary because they prime transcription of viral messenger RNA from the virion encapsulated RNAs, or the vRNAs. An ambisense genome is a genome in which both nucleic acid strands encode for proteins. This is seen for the S segment of the flevovirus and the S and M segments of the tospovirus. All of the Arenaviridae, which is the next family we will discuss, have ambisense orientation. So don't stress out too much about it. This is sort of like an introduction to it, because the most important groups that we're going to talk about for the Bunyaviridae do not have ambisense RNA. But basically what you see in this illustration is the comparative replication schemes of typical negative sense genome orientation and ambisense genome. So if you look at the left side, that's the negative sense genome segment. That's what we normally have talked about. So you have the genome segment in the middle. Remember, this is minus sense RNA. And then the antigenome, which is complementary to the genome segment, is just used for replication, making more full-length copies of the genome. And you have transcription of messenger RNA that occurs from the full-length minus strand genome. So that's what we've normally talked about with all of these minus sense RNA viruses. With ambisense, what happens is that one half of the segment is has one polarity and the other half is different. So if you look at the right hand side, what you have here is a minus strand genome, which is in the middle, and full-length copies of that are replicated by transcription, but you have messenger RNA being transcribed from the 3' end only of the negative strand genomic RNA. Then when you make your complementary full-length strand, the 3' end of that is going to be transcribed into messenger. So this is a way of controlling the production of viral proteins. And the concentration of the N protein plays a role in controlling this, because remember, the full-length copies are going to be encapsulated with the N protein, and only the messenger RNAs will not have the M protein associated with it. Now, when we look at the translation and processing of viral proteins, remember there are 3 segments. The small segment carries the N protein, the nucleocapsid protein, and a small non-structural protein. These are translated on free ribosomes. The M segment carries the 2 glycoproteins, GC and GN, and another non-structural protein. These are translated on membrane-bound ribosomes because remember, glycoproteins are going to have to be processed through the Golgi to become glycosylated, and that's how they become inserted into the membrane to form the envelope for the virus. So again, the sort of thing that we've seen before. And then the L segment carries the polymerase gene, and the polymerase is translated on free ribosomes. Again, as you would expect, it's not going to be glycosylated. Here you can see the assembly and release of virions. There's a direct interaction between the RNPs and the envelope proteins. This particular group of viruses does not have a matrix protein, so it's a direct interaction. Most budding is into the cisternae of the Golgi apparatus. And then the virions are going to be transported to the cell surface in vesicles. And so we have migration of Golgi vesicles containing viruses to the cell surface, fusion of the vesicle membranes with the plasma membrane, resulting in release of infectious viruses. Some viruses in certain cell types can bud both into intracellular vesicles and also from the plasma membrane, but most often it's budding through intracellular membranes. And here's just a review of the entire replication cycle. So, pretty typical in terms of attachment and entry and uncoding. So the surface glycoproteins bind with receptors. It's brought in through endocytosis. Acidification of the endosome results in uncoding and release of the RNPs into the cytoplasm. And messenger RNA can be produced. The glycoproteins will be translated at the ER membrane. Then the other proteins are going to be directly translated just in the cytoplasm. You're going to go through a complementary RNA strand to make more viral RNA, and that's going to bind to the glycoproteins that are inserted into the Golgi membrane. Then you're going to have the virus budding into the Golgi, being released up to the plasma membrane in vesicles. And then the vesicle will fuse, I should say, with the plasma membrane, and the virion will be released. The members of the Bunyaviridae family are collectively called bunyaviruses, and they make up the largest family of viruses with an RNA genome. The Bunyaviridae family is the largest group of arboviruses, or arthropod-borne viruses, with over 350 members distributed worldwide. The family comprises 5 genera: Cospovirus, Hantavirus, Norovirus, Orthobunyavirus, and Flebovirus. With the exception of the hantaviruses and the tospoviruses, all other bunyaviruses are arboviruses whose natural life cycle involves replication in arthropods such as mosquitoes, sandflies, and ticks, and also vertebrates. So these viruses will alternate between an insect host and a vertebrate host. Although tospoviruses are also transmitted by arthropods, in this case a thrip species, their natural life cycle doesn't involve any vertebrate replication. Instead, they are plant pathogens that can cause significant worldwide crop losses. The odd one out would be the hantaviruses, which can infect vertebrates, but they're rodent-borne rather than being transmitted by insects. They're typically transmitted to humans through infected excretions and secretions. Let's consider a couple of notable examples of viruses in the Bunyaviridae. Let's start with lacrosse virus. This is my favorite virus because it's the virus that I've worked with since I started my PhD more years ago than I care to admit. In the United States, approximately 80 to 100 lacrosse encephalitis virus neuroinvasive disease cases are reported each year. That may sound like a small number, but it's a significant underdiagnosis and underreporting of disease, because typically it's only the most severe manifestations of the disease, with patients demonstrating central nervous system infection, that you actually get a diagnosis. Less severe infection generally results in a febrile illness that doesn't cause encephalitis. And patients who develop those kinds of infections, oftentimes they just think that they have a summer cold. But what we found over the years of research is that in endemic areas, the number of people who have been infected with lacrosse can actually be quite a substantial portion of the population. So for instance, in Indiana, one of my professors at Notre Dame had gained access to the supply of, a freezer full of blood samples that had been taken from people who were getting their prenuptial blood tests done in order to get their marriage license. Back in the old days, people had to go in and get their blood tested, and in order to be screened for STDs before they could obtain a marriage license. So the state had this whole big supply of these blood samples taken from people over a number of years from all over the state of Indiana. And when he did serological testing, what he found was that the overall seropositivity rate for people in that area of the country was about 5%. So that translates to thousands and thousands of actual cases of lacrosse. Now, when we look at the disease cycle, this is why I like to work with this virus, because it has such a complex and interesting cycle. So we have, of course, horizontal transmission. That's the blue circle down at the bottom. And the virus normally circulates between mosquitoes and small woodland mammals, such as chipmunks and squirrels. Those act as the amplifying host. So a mosquito will come in and feed from a chipmunk, transmit the virus, then that chipmunk will become viremic, and other uninfected mosquitoes, when they feed on that chipmunk, will acquire the virus. And that's the basic horizontal cycle. People are considered dead-end hosts. And remember, a dead-end host doesn't mean that the host dies. It just means that that host doesn't transmit the virus from them. So in the case of humans, generally they don't produce a high enough circulating viremia so that more mosquitoes, when they feed on these individuals, will become infected. Now we have a transovarial cycle as well, vertical transmission. An infected female can transmit the virus to her progeny through the egg. And when those eggs hatch, the larvae will be infected and they will give rise to infected adults. This is important because this is the overwintering mechanism for the virus as well as for the mosquito. The mosquito lays drought-resistant eggs that are freeze-resistant, and the infected larva inside the egg survives so that when it hatches in the spring, it emerges as an infected adult. Now, an interesting aspect of transovarial transmission is that while through horizontal transmission only females can become infected because they have to take a blood meal, when you're talking about transovarial infection, both males and females become infected. And that leads to another type of horizontal transmission, which is male to female through venereal transmission. So a male that emerged infected through transovarial transmission can then transfer the virus to an uninfected female. So we have all different kinds of modes of infection, plus the typical enzootic areas where this virus occurs are nice, beautiful woodlands. I don't have to go to swamps and places like that. Instead, I can go to the national forest to do my research. Now let's consider the hantaviruses. Remember, I said that they were different from the other bunyaviruses because they use a rodent as a vector as opposed to an insect. The first hantavirus described was Korean hemorrhagic fever. It occurred in U.S. troops during the Korean War. During that war, over 3,000 cases of febrile disease with hemorrhagic and renal manifestations was recorded, and eventually the virus was identified. There was some research done on the old world hantaviruses, but the hantaviruses really didn't come back into importance until there was an outbreak in the Four Corners area of the southwest United States in 1993. And that created a lot of interest again in these viruses. And we found some interesting data that allows us to separate the hantaviruses into Old World hantaviruses and New World hantaviruses. There's phylogenetic analysis of Old World and American hantaviruses indicates that the relationship among hantaviruses corresponds with the phylogeny of the rodent hosts. The apparent coevolution of hantaviruses with their reservoir hosts is evident by the commonly observed close association of each hantavirus species with a certain rodent species. In addition, phylogenetic analysis of hantavirus sequences and mitochondrial sequences from their rodent hosts suggest a long-standing coevolutionary history of hantaviruses with their rodent carriers. This distinction can narrow the search for a rodent host for a newly discovered HPS-like disease and suggest disease implications for various new viruses being genetically amplified from rodents. National surveillance for hantavirus infections in the United States began in 1993 during an outbreak of severe respiratory illness in the Four Corners region, and hantavirus pulmonary syndrome, or HPS, became nationally notifiable in 1995. The presence of fever and pulmonary symptoms in a patient with laboratory-confirmed evidence of hantavirus infection is required for an HPS case to be reported through the Nationally Notifiable Diseases Surveillance System. In 2014, the Council of State and Territorial Epidemiologists resolved to expand the national reporting of laboratory-confirmed hantavirus infections to include HPS and non-pulmonary hantavirus infection. The first year of reporting of non-pulmonary hantavirus cases was 2015. As of January 2017, a total of 728 cases of hantavirus infection have been reported in the United States. Of persons ill with HPS, 63% have been male, 37% female. The mean age of confirmed case patients is 38 years old, with a range of 5 to 84 years. And cases have been reported in 36 states. More than 96% of the cases have occurred in states west of the Mississippi River. So the data that's available from CDC is a little bit dated. It's up through January 2017, and they haven't updated anything since then. But the pattern hasn't changed. The numbers on the states have undoubtedly altered a bit over the past couple of years, but the pattern hasn't changed. Several hantaviruses that are pathogenic for humans have been identified in the United States. In general, each virus has a single primary rodent host. Other small mammals can be infected as well, but are much less likely to transmit the virus to other animals or humans. The deer mouse is the host for Cynombre virus, or SNV, the primary causative agent of HPS in the United States. The deer mouse is most common and widespread in rural areas throughout much of the U.S. Although prevalence varies temporally and geographically, on average, about 10% of deer mice tested throughout the range of the species show evidence of infection with SNV. Cynombre virus has an interesting story behind its name. Viruses are generally named for the place where they were identified. So La Crosse virus, for instance, was first identified in La Crosse, Wisconsin. Cynombre is Spanish for no name. It got its name from the fact that nobody wanted to be associated with hantavirus. When the outbreak occurred in '93, there was a lot of press coverage, and people started getting worried about the virus. And the states where the virus was popping up were concerned about tourism. Nobody could agree on what to call the virus, so eventually one of the virologists threw up his hands and just said, we'll call it no-name virus, or sin nombre. The rodent hosts of HPS are generally not associated with urban environments, although some species, including the deer mouse and the white-footed mouse, will enter human habitation in rural and suburban areas. So with this, again, remember, it's the rodent host that's responsible for transmission. Hantaviruses do not cause overt illness in the reservoir hosts. Although infected rodents shed virus in saliva, urine, and feces for many weeks or months, or even for life, the quantity of virus shed can be at its greatest approximately 3 to 8 weeks after infection. Field data suggests that transmission in host populations occurs horizontally, and that this occurs more frequently among male than female rodents. Transmission from rodent to rodent is believed to occur primarily after weaning and through physical contact, perhaps through aggressive behavior such as fighting. Studies of the genomic sequences indicate that the virus has probably evolved concurrently with its rodent host over a long period of time. Occasional evidence of infection is found in numerous other species of rodents and their predators, such as dogs and cats, indicating that many, perhaps any, mammal species coming into contact with an infected host might become infected. No evidence supports the transmission of infection to other animals or to humans from these dead-end hosts. However, domestic cats and dogs may bring infected rodents into contact with humans. Ticks, fleas, mosquitoes, and other biting insects have not been implicated in the transmission of hantavirus pulmonary syndrome. Human infection occurs most commonly through the inhalation of infectious aerosolized saliva or excreta. Persons visiting laboratories where infected rodents were housed have been infected after only a few minutes of exposure to animal holding areas. Transmission can occur when dried materials contaminated by rodent excreta are disturbed and inhaled, directly introduced into broken skin or conjunctiva, or possibly when ingested in contaminated food or water. Persons have also acquired HPS after being bitten by rodents. High risk of exposure has been associated with entering and cleaning rodent-infested structures. Person-to-person transmission has not been associated with HPS cases in the United States. However, person-to-person transmission, including nosocomial transmission of Andes virus, was well documented for a single outbreak in southern Argentina, and it was suspected to have occurred much less extensively in another outbreak in Chile that was associated with the same virus. Therefore, universal precautions are always recommended for healthcare workers treating HPS patients. Once the hantavirus enters the human body, it replicates and spreads. In the lungs, the virus causes blood vessels to become weak and leaky, which fills air sacs with blood and makes breathing difficult. In the heart, damage to the heart muscle itself, plus the weak and leaky blood vessels, reduce the heart's ability to pump oxygen-filled blood to organs and cells of the body. Without oxygen and adequate blood pressure, the body can go into shock, which in turn can result in organ failure followed rapidly by death. The first stage of the disease is the incubation phase. It produces no outward signs. This phase can last for up to 8 weeks after exposure to the virus. The second phase of early HPS symptoms develop quickly. This phase lasts from 2 to 8 days. In addition to fever and myalgias, early symptoms include headache, chills, dizziness, non-productive cough, nausea, vomiting, and other gastrointestinal symptoms. Malaise, diarrhea, and lightheadedness are reported by approximately half of all patients, with less frequent reports of arthralgias, back pain, and abdominal pain. Patients may report shortness of breath. However, the diagnosis is seldom made at this stage. It's just too nondescript. Once the cardiopulmonary phase begins, however, which includes coughing and tachypnea, which is very, very rapid breathing, the disease progresses rapidly, necessitating hospitalization and often ventilation. Normal lung tissue is airy, almost fluffy. It becomes increasingly heavy and it demands more and more effort for every breath as fluid from blood is seeping in. It seeps out into the air sacs, making it harder for the patient to get oxygen. Quarts of fluid can accumulate in the chest cavity. A normal lung is whitish and like a soft sponge. Lungs from patients are dark bluish red from lack of oxygen and cut like meat. You'll hear that expression, cuts like meat, in the hot zone. In x-rays, you get what's called a whiteout. So look at these pictures here. This is a progression over the course of just a couple of days for the same patient. On a normal chest film, you should see a black lung field with white ribs and grayish lung markings. But with lungs that are full of fluid, the radiological density is increased and it starts to look like a snowstorm. So you can see in the first film up at the top left, the patient has part of the lung is showing the whiteout. So he has reduced lung capacity, but still a lot of the lung is normal. The upper parts where it's dark, you can see he's still able to get breath from there. But after less than 24 hours, you can see he's down to just a third of his normal capacity. And by the last film, you can see that almost the entire lung field now is showing that whiteout kind of phenomenon. And unfortunately, this patient died soon after. Now, in terms of precautions, it's pretty common sense sort of things. The virus is normally picked up from exposure to rats and their excrements, their droppings, and so on. So you want to do things that keep rats out of your home and workplaces, your storage buildings, and so on. You want to remove brush and wood piles and things, anything that would attract rats and mice. Close garbage containers and things. But what's important is the care that you should take if you go into an area where there could be mouse droppings. Before cleaning up nests or droppings that are found inside a building, whether it's a home or a trailer or a storage building, whatever, you want to open windows and doors to ventilate the area for at least 30 minutes. If you're going to be stirring up dust, you should wear goggles and a HEPA or N95 mask. You want to wear protective gloves to handle the dead mice or rats that might be there. You want to avoid stirring up dust by sweeping or vacuuming. If that can't be avoided, then you want to dampen the areas before cleanup by just spraying a 1 to 10 bleach water solution or other kind of disinfectant and let that sit for a bit. That will not only inactivate the virus, but it will eliminate stirring up dust. After 30 minutes, put on your viricide again and then begin the cleaning process. So, you just want to avoid doing anything that can attract rats and mice in to an area and take precautions when you're cleaning up. Precautions for hikers and campers would include things, obviously you want to avoid contact with the rodents. You need to disinfect, clean out cabins and shelters before you use them. You want to be sure that you're sleeping in an area that's not close to where rodents are living. Um, you want to keep your food in rodent-proof containers, and so on. So various kinds of precautions can be taken. I remember several years ago, on a weekend morning, I was out walking my dog over in the wooded area at the end of Plantation Road. And a young man came out from the woods there, and we started talking. It turned out he was a graduate student in the wildlife department, and he was practicing with his traps. He was going to be doing small animal trapping out in West Virginia. And we had a nice talk and so on. I found out about a year later that he had died from hantavirus. We don't know now whether he might have gotten infected through handling the traps, or if he got infected in the fairly primitive cabin where he was staying. But nevertheless, it really brought home to me, the risks that we have even around here in this part of the country. So now Virginia Tech has very strict guidelines for anybody who's going to be doing small animal trapping like that, which includes the necessity of wearing the N95s and protective gloves and so on. There was an outbreak of hantavirus in visitors to Yosemite National Park in 2012. The NPS public health officials believe that the people became infected while staying at the Signature Tent Cabins in Curry Village in Yosemite National Park. So they did very careful epidemiological studies and tracked down the people. The timeline you can see here of when people visited Yosemite, and the star at the end of the line indicates when they had onset of illness. And remember that the incubation period can be quite extended with these viruses. But they had 10 cases, 5 of which required intensive care, and 3 of which, or 30% of the cases, died. Here you can see what those signature tent cabins look like. They found that rodent nests and tunnels were being formed in the foam insulation of the cabin walls, which were covered up normally by the sides of the tent. Rodent trapping in the implicated area resulted in high trap success, and antibodies reactive to Cynombre virus were detected in 14% of the captured deer mice. All the signature tent cabins were closed and subsequently dismantled. But continuous public awareness and rodent control and exclusion are key measures in minimizing the risk for hantavirus infection in areas inhabited by deer mice. Here's an interesting outbreak of a hantavirus. In this case, it was Seoul virus, Seoul as in Seoul, Korea. The CDC first identified cases of pet rat to human transmission of Seoul virus in late 2016 when they were notified of local cases from health officials in Tennessee and Wisconsin. According to the report, one patient who was hospitalized with influenza-like symptoms such as fever and low white blood cell count owned and operated an in-home rat breeding facility, or a rattery, with approximately 100 Norway rats, which happens to be the primary host of the virus. This spurred a larger investigation in which researchers identified the 31 U.S. sites, which included ratteries, homes, and pet stores, with cases of Seoul virus infection. 6 of those locations reported exchanging rats with Canadian ratteries. Investigators then tested blood supplies from 183 people in the U.S. and Canada and found that 18 individuals had signs of a recent infection. 3 people had been hospitalized with infection-related illness, but no deaths occurred. Symptoms of sole virus infection range from mild influenza-like illness to hemorrhagic fever with renal syndrome, which can cause acute kidney failure and be fatal. According to the CDC, the virus is not known to spread between people. But the bottom line, the take-home message, is that pet rat owners should practice safe rodent handling to prevent sole virus infection. Okay, so let's now move on to our next virus family, the Arenaviridae. The Arenaviridae are a family of viruses whose members are generally associated with rodent-transmitted diseases in humans. Each virus is usually associated with a particular host species in which it is maintained, so very similar to what we saw with the hantaviruses. Arenavirus infections are relatively common in humans in some areas of the world and can cause severe illness. The particles are spherical and enveloped. When viewed in cross-section, they show grainy particles that are actually ribosomes acquired from their host cells. It is this characteristic that gave them their name, derived from the Latin word arena, which means sandy. Their genome is composed of RNA only, and the replication strategy is kind of complicated, but don't worry, we'll talk about it. The viral envelope contains multiple copies of glycoprotein spikes on the surface that are required for receptor binding and virus entry. The Z protein forms a matrix layer underneath the viral membrane. The nucleoprotein NP associates with the polymerase L to form, along with the genomic RNA, the ribonucleoprotein or RNP complex. Arenaviruses contain a bisegmented negative strand genome composed of the small RNA segment and the large RNA segment. Each RNA segment encodes two viral proteins in ambisense orientation, and we'll go back over what ambisense orientation means in a couple of slides. The open reading frames are separated by intergenic regions with hairpin loops. The virions are spherical or pleomorphic with dense lipid envelopes. The virion surface is covered with the club-shaped glycoprotein spikes that have a distinctive stalk and head regions. These spikes are made of trimeric spike structures of two virus-encoded membrane glycoproteins, the GP1 and GP2. Isolated RNP complexes are organized into beads on a string-like structures. The virus is capable of infecting many different cell types from many different species. The receptor binding is mediated by GP1. Remember, the spikes are composed of GP1 and GP2. So what happens here is the mature GP trimer covers the virion surface, or we virologists like to say decorates the virion surface, and binds with cellular receptors. Under acidic pH, GP1 dissociates and liberates the fusion peptide of GP2, triggering fusion between the viral and cellular membrane. So this is just another example of how low pH is required to have the change that allows for fusion. And then with fusion, you get release of the RNPs into the cytoplasm for uncoding. Okay, so now let's look at the genome organization of the arenavirus. The genomes consist of two segments of RNA, a large and a small, and both of these are ambisense. The S segment corresponds to the bunyavirus S and M segments linked tail to tail in an ambisense arrangement. So if you think back to the genome organization that we talked about for most of the bunyaviruses, they had a small segment that coded for N, the middle segment, the mid-sized segment that coded for the glycoproteins, and then the large segment that coded for the polymerase. So the smaller segment here for the arenaviruses is as if you took those two bunyavirus segments, the S and the M, and bound them together, but in an ambisense organization. The L segment corresponds to the L segment of the bunyavirus with the addition of a second gene, Z, which is a non-structural protein in ambisense orientation. So this is the type of organization that we see. Now let's look at how it works. So again, it's ambisense. This provides a mechanism for temporal regulation of gene expression. That's something that different viruses control in various ways. And this is one way of doing it, although I would argue kind of a complicated way, but it does work. So what we have is the mRNA of one gene is synthesized from the genomic RNA and is expressed early, whereas the mRNA for the second gene is synthesized from the antigenomic RNA and is expressed late. As in bunyaviruses, synthesis of arenavirus mRNA occurs in the cytoplasm using a primer that is snatched from cellular mRNA. So we have cap snatching in this virus family as well. And there's a secondary structure in the RNA between the two ambisense genes that causes termination of transcription, and the mRNAs are not polyadenylated. The genomic sRNA is a template for the synthesis of messenger RNA for N, and N is therefore expressed early after infection. Because N is required for the replication of the virtual RNA, as for all negative strand RNA viruses, this arrangement is necessary if the virus is to replicate. The mRNA for G1 and G2 is transcribed from the antigenomic copy of S and is therefore expressed late. The glycoproteins are produced as a polyprotein that is cleaved. Producing glycoproteins late has the effect of delaying virus assembly. Now let's look more closely at how this works for timing protein expression and RNA replication. Replication of the viral genome occurs in the cytoplasm following its release from the endosome. The viral NP and L proteins are the minimum known components required for viral genome replication. Preliminary evidence suggests that the timing of viral RNA replication is under regimented control mechanisms. NP and L genes are transcribed and translated first and directly from the negative sense viral genome. GPC and Z are expressed several hours later after the viral genomic RNA segments encoding these genes have been replicated and then transcribed. So this hopefully is starting to make a little bit of sense. The NP protein and the L protein are needed first for the replication cycle of the virus. Remember, the NP is what encapsulates the RNA, and the L is the polymerase, which is necessary to replicate the RNA. So these two genes are translated from messenger that's produced directly from the genome RNA, that is the negative full-length strand RNA. Then after you've made complementary copies of that RNA, the GPC and Z proteins are translated from messenger that's transcribed from that positive sense RNA. So it's a way of controlling the timing of things and delaying the formation of intact virions. GPC is transported through the endoplasmic reticulum and Golgi compartments. Translated NP, L, and Z are transported to the platform for virus budding, gathered together with viral RNPs, and cleave GP, and then pinched off from the surface of infected cells. Taking the model for regulation of arenavirus RNA synthesis by Z together with the results of NMR analysis, it is proposed that at the early stage of arenavirus replication, when Z is present at low concentration, Z permits ongoing RNA synthesis. Once Z reaches a high concentration, the binding of Z to RNP results in inhibition of viral transcription and replication. Transportation of Z to the cellular membrane, the endosomal membrane or the plasma membrane, is not known well. Studies suggest that assembly occurs after attaching to the plasma membrane. Therefore, Z might interact with NP and L, which encapsulate the viral RNA to compose the viral RNP complex before inactivation, or I should say before interaction with the plasma membrane. So here's the entire replication cycle. Virus attaches to host receptors through GP glycoprotein and is brought in by endocytosis into vesicles in the host cell. Fusion of the virus membrane with the vesicle membrane occurs. The ribonucleic capsid is released into the cytoplasm. You get sequential transcription. Viral messenger RNAs are capped in the cytoplasm. Replication presumably starts when enough nucleoprotein is present to encapsulate the newly synthesized antigenomes and their genomes. This should sound familiar. This is the kind of process that we've talked about with all of these minus-sense RNAs. The ribonucleic capsid interacts with the Z protein under the plasma membrane and buds, releasing the complete virus virion. Now let's look at some of the arenaviruses that are pathogenic to humans. The Old World and New World subgroups are polyphyletic and contain both human pathogenic and non-pathogenic viral strains, with 10 strains in total known to cause human diseases. Unlike other hemorrhagic fever viruses, such as the Ebola virus, arenaviral transmissions to humans have been found primarily as a result of human interactions with the rodents as the natural reservoirs of these viruses. However, human-to-human transmission may play a larger role in certain viral outbreaks. The natural history of the arenaviruses is very similar to that of the hantaviruses. They establish a persistent infection in a single rodent host. Many cause hemorrhagic disease in man following transmission by aerosolization of virus excreted in urine or feces. Like hantavirus, they appear to have co-evolved with their hosts. In both, an evolutionary tree of the viruses resembles the tree that describes their rodent hosts. Many similarities in genome organization and expression, the association with a single rodent species, and the nature of the disease in man suggest that the arenaviruses are closely related to the hantaviruses. A reasonable hypothesis is that the arenaviruses arose from the hantaviruses by fusion of the S and M segments to form one segment, which allowed for finer control of the virus life cycle. These viruses are zoonotic, meaning that in nature they are found in animals. Each virus is associated with either one species or a few closely related rodents, which constitute the virus's natural reservoir. There are several different diseases associated with the arenaviruses. We have the Old World, New World, and then LCMV, which is cosmopolitan, meaning it has a worldwide distribution, although it most likely evolved originally as an Old World virus. Let's look at the epidemiology. With these viruses, you have rodent to human transmission. So it's through aerosolization, ingestion of contaminated food, and catching and preparing infected rodents as a food source. So very much like what we saw with hantaviruses. The big difference here is that human to human transmission does occur and can lead to increased spread of the disease. This occurs when you have direct contact with blood, tissues, and secretions of infected humans. A needle stick or cut can lead to infection. Inhalation of aerosolized virus can occur. So if there's a patient suffering from the hemorrhagic disease, they're bleeding profusely, the blood dries, and then when it's being cleaned up, some of it is aerosolized. That's a way for human to human transmission to occur. But as you can see, caregivers and healthcare people are at special risk for contracting Lassa fever and diseases in this group. Let's look at lymphocytic choriomeningitis first, LCMV. LCMV infections can occur after exposure to fresh urine, droppings, saliva, or nesting materials from infected rodents. Transmission may also occur when these materials are directly introduced into broken skin, the nose, the eyes, or the mouth, or presumably by the bite of an infected rodent. Person-to-person transmission has not been reported, with the exception of vertical transmission from infected mother to fetus, and rarely through organ transplantation. Individuals of all ages who come into contact with the infected excrement and saliva and blood of wild mice are potentially at risk for infection. Owners of pet mice or hamsters may be at risk of infection if these animals originate from colonies that were contaminated with the virus, or if their animals are infected from other wild mice. Human fetuses are at risk of acquiring infection vertically from an infected mother. Laboratory workers who work with the virus or handle infected animals are also at risk. However, this risk can be minimized by utilizing animals from sources that regularly test for the virus, wearing proper protective laboratory gear, and following appropriate safety precautions. In May 2005, 4 solid organ transplant recipients contracted an illness that was later diagnosed as lymphocytic choriomeningitis. All received organs from a common donor, and within a month of transplantation, 3 of the 4 recipients had died as a result of viral infection. Epidemiologic investigation traced the source to a pet hamster that the organ donor had recently purchased from a Rhode Island pet store. Similar cases occurred in Massachusetts in 2008 and in Australia in 2013. Currently, there is not an LCMV infection test that is approved by the Food and Drug Administration for organ donor screening. The Morbidity and Mortality Weekly Report advises healthcare providers to consider LCMV infection in patients with aseptic meningitis and encephalitis, and in organ transplant recipients with unexplained fever, hepatitis, or multisystem organ failure. LCMV infection usually is either asymptomatic or causes a mild self-limited illness in otherwise healthy people. But it can cause aseptic meningitis, although these infections are rarely fatal. Infection during pregnancy can result in vertical transmission of the virus from mother to fetus. LCMV infection during the first or second trimesters can lead to severe illness in the fetus. Serological studies conducted in urban areas of the United States have indicated that the prevalence of LCMV infections amongst humans is approximately 5%. The house mouse is the primary reservoir for the virus, with a prevalence of infection of anywhere from 3 to 40%. A high degree of focality is often noted. However, other types of rodents, such as hamsters or guinea pigs, can be infected after contact with infected house mice. These rodents have also been implicated in human infection. Animals can become ill or can be asymptomatic. Infections in humans occurs primarily through exposure to secretions or excretions of infected animals. How to reduce the risk of LCMV? Well, got to wash your hands with soap and water after handling pet rodents, cleaning cages. You have to keep the rodent cages clean and free of soiled bedding. Clean the cage in a well-vented area or outside. And of course, make sure that wild mice don't have access to the food or bedding or the cages of your animals. And last but not least, I don't care how much your hamster smiles at you, like this little guy in the picture here, do not kiss your pet rodents or hold them close to your face. Lassa virus is an old world arenavirus. It's the etiologic agent of Lassa fever, a severe human disease that is reported in more than 100,000 patients annually in the endemic regions of West Africa. Mortality rates for hospitalized patients varies between 5 and 10%. Currently, there's no approved vaccines against Lassa fever for use in humans. So you just have various kinds of supportive care, such as making sure that the patient doesn't become dehydrated and things like that. But this is a disease that you have to be especially careful about the possibility of nosocomial transmission. Here we see Lassa fever outbreaks, an estimated 100,000 to 300,000 cases occur annually in West Africa, with 5,000 deaths estimated. So, you can see that it's definitely an enzootic disease that's pretty well established. Here you can see Lassa fever cases over time. The numbers of clinically suspected and confirmed cases of Lassa fever identified are at one particular teaching hospital are shown in this graph starting in January 2012 through May 2018. Gray shading indicates the typical dry season in this area. This is in Nigeria, and it overlaps with the period of peak Lassa fever incidence. During 2018, there was an unusual increase in Lassa fever cases occurring in Nigeria that raised concerns amongst national and international public health agencies. They looked at virus genomes from over 200 patients, and they found that the increase in cases was not attributable to a particular Lassa virus strain or sustained by human-to-human transmission. Instead, the data were consistent with ongoing cross-species transmission from local rodent populations. Phylogenetic analysis also revealed extensive viral diversity that was structured according to geography, with major rivers appearing to act as barriers to the migration of the rodent reservoir. There was an outbreak of Lassa fever in Nigeria in 2020 that was a bit larger than previous years. If you look at that graph in the very bottom left, you can see the different years, and the red line indicates the Lassa fever activity for 2020. So the outbreak began in January. At the end of April, the outbreak was declared over. The Lassa fever emergency was over after there was a steep decline in cases. And of course, you had a number of different agencies that worked very hard to bring this under control. Now, while the Lassa fever emergency has subsided for 2020, it remains an endemic disease and will probably continue to crop up in future years. You can see that is pretty clear from that graph that I just directed you to. One thing that was different during this outbreak compared to others is that they did a much better job of protecting healthcare workers. The protection of healthcare workers represents a substantial step forward in screening, testing, and treating Lassa fever in Nigeria. In 2018, during that outbreak that we mentioned in the previous slide, this was one of the primary goals established. By protecting healthcare workers, more patients can be treated, and hospitals do not become amplifiers of the virus. Signs and symptoms of Lassa fever typically occur 1 to 3 weeks after the patient comes into contact with the virus. For the majority of Lassa fever virus infections, approximately 80%, symptoms are mild and undiagnosed. Mild symptoms include slight fever, general malaise and weakness, and headache. In 20% of infected individuals, however, disease may progress to more serious symptoms, including hemorrhaging, such as from the gums, the eyes, and the nose. Also, respiratory distress, vomiting, facial swelling, pain in the chest, back, and abdomen, and the patient may even go into shock. Neurological problems have also been described, including hearing loss, tremors, and encephalitis. Death may occur within 2 weeks after symptom onset due to multi-organ failure. The most common complication of Lassa fever is deafness. Various degrees of deafness occur in approximately 1/3 of infections, and in many cases, hearing loss is permanent. As far as is known, the severity of the disease does not affect this complication. The deafness can occur even in mild cases, not just in the severe cases. Approximately 15 to 20% of patients hospitalized for Lassa fever die from the illness. However, only about 1% of all Lassa virus infections result in death. Death rates for women in the 3rd trimester of pregnancy are particularly high. Spontaneous abortion or miscarriage is a serious complication of infection with an estimated 95% mortality in fetuses of infected pregnant mothers. Because the symptoms of Lassa fever are so varied and nonspecific, clinical diagnosis is often difficult. Lassa fever is also associated with occasional epidemics during which the case fatality rate can reach as high as 50% in hospitalized patients. Now, prevention and control. The best mechanism that they found is village-based programs for rodent control and avoidance. Now, if you look at the slide, you see up at the top right, there are 2 people that they sort of look like witch doctors or whatever, and they're not witch doctors. The people know exactly how this virus is transmitted, but keep in mind that most of this population is illiterate. So you can make all the brochures that you want. The people won't be able to read them. So as part of these village-based programs, they have people that go out and put on little plays that demonstrate to the folks how to avoid coming into contact with Lhasa. And it becomes a fun thing. You know, the kids come and they learn something and so on. So it's a way of getting around the fact that you have a large portion of the population that can't read. Then, of course, hospital training programs. You want to avoid nosocomial spread. We mentioned that that was a big step forward that they saw in the 2020 outbreak in Nigeria. They need better diagnostic tools, too. Now, there is an antiviral chemotherapy that may help, ribavirin. Ribavirin seems to help if given early in infection. It's not a cure. And there's no evidence that ribavirin can be effective as a prophylactic. And in addition to the village-based programs with the actors and so on, of course they do put out information, and there's a lot of illustrations, cartoons to go along with it. I like this one down at the bottom because in addition to things like washing your hands and storing your food in rodent-proof containers and so on, it recommends that you keep a cat. Now, how practical is that? Okay, the next and last virus family for this lecture would be the orthomyxoviridae. The influenza virion is roughly spherical. It is an envelope virus. Inserted into the lipid membrane are the HA, hemagglutinin, and NA, neuraminidase glycoproteins. The HA and NA are important in the immune response against the virus. Beneath the envelope membrane is a viral protein called M1, or matrix protein. This protein gives strength and rigidity to the lipid envelope. Within the interior of the virion are the viral RNAs, 8 of them. These are the genetic material of the virus, and they each code for 1 or 2 proteins. The influenza viruses have a negative polarity RNA genome fragmented into 8 single-stranded molecules. These are never present as naked RNA in virus particles or infected cells, but exist as ribonucleoprotein complexes, or RNPs, generated by interaction of each RNA segment with the viral polymerase and a number of nucleoprotein, or NP, molecules depending on the RNA length. After the virion attaches to sialic acid-containing receptors at the cell surface, the virus receptor complex is taken into cells by endocytosis. As the endosomal vesicles that contain the virus particles move towards the cell nucleus, their pH drops. This change in pH is accomplished by a cellular channel that pumps protons, or hydrogen ions, into the vesicle. When the endosomal pH reaches 5.0, the viral HA protein undergoes a conformational rearrangement. This change exposes a fusion peptide on the HA, a short hydrophobic sequence that inserts into the endosomal membrane and causes it to fuse with the viral envelope. When this occurs, the viral RNAs are released into the cytoplasm. They are then transported into the cell nucleus where viral RNA replication occurs. In the influenza virion, the viral RNAs are not naked, but bound to a number of viral proteins, including the M1 protein. This protein forms the shell that underlies the lipid membrane of the virion, as I mentioned. Unfortunately, if the viral RNAs are bound to M1 protein when they are released from the virion, they cannot enter the nucleus. To get around this problem, the influenza virion has a few copies of it in its membrane of a protein called M2. This viral protein forms a channel in the membrane that actively pumps protons from the endosome into the interior of the virion. These protons lower the pH in the interior of the virion, releasing the viral RNAs from N1. In this way, the RNAs can enter the nucleus. So let's take another look at that. Okay, so let's look at this a little more closely. The M2 protein forms ion channels in membranes that allow passage of hydrogen ions or protons. To become active, the polymerase in the interior of the virus must be exposed to low pH. Influenza enters the cell in endosomes that are progressively acidified. The acidic pH not only triggers a conformational change in the HA that results in fusion of the viral membrane with the endosomal membrane, but it also activates the RNA polymerase of the virion through the activity of M2. So this is an extra step. You have the interior of the virion also becoming acidified, not just the external part of the endosome around it. So it's like a two-step process of acidification. When we look at transcription and translation, the complementary RNAs are exact complements of the genomic minus strands, full-length copies. Influenza has a polymerase complex composed of 3 protein subunits, and you can see that on the bottom left of the slide. You have the polymerase basic protein 1, or PB1, the polymerase basic protein 2, or PB2, and polymerase acidic protein, or PA. All 3 components are essential for viral replication. The way influenza replicates is that the PB2 subunit binds to the host cellular pre-messenger RNA and is cleaved by the endonuclease in the PA subunit. The process allows for the transcription of messenger RNA by the RNA polymerase function of PB1. Essentially, we're talking about cap snatching. The active forms of viral RNAs are the RNPs, and each one acts as an independent functional unit during transcription and replication. This property is in line with the high frequency of genetic reassortment amongst influenza viruses in double infections. Likewise, the end product of RNP replication is not a naked RNA, but a new RNP. When sufficient amounts of viral protein have been synthesized and transported to the nucleus, viral RNA replication begins. During replication, the viral genome is copied into a faithful antigenomic RNA, or vcRNA, which is a perfect complement of the genome and serves as a template for the production of genomic RNA, or vRNA. The biochemical basis for the switch is not known, but the NP protein undoubtedly plays a role. So during the course of infection, messenger RNA synthesis occurs before cRNA and vRNA transcription, and messenger RNA transcription is much more abundant because the use of primers significantly increases the initiation efficiency. Remember, the primers come from cap snatching. The initial mRNAs are transcribed by the vRNP-associated polymerases and are exported from the nucleus for translation by cytoplasmic ribosomes. Inside the nucleus, the replication of the influenza genome involves 2 steps: transcription of complementary RNA, or the cRNA, followed by transcription of new vRNA copies using the cRNAs as templates. The cRNAs are produced by an unprimed process that relies on the correct complementation of free are NTPs with the 3' end of the vRNA template. Upon exiting the polymerase, the cRNA associates with newly synthesized NP molecules and a single copy of the viral polymerase to assemble into a cRNP. And then the cRNP can act as the basis for transcription of the vRNP. Budding must result in the packaging of the 8 different genomic segments that constitute the viral genome into one virus particle if it's going to be infectious. The virus appears to package more than 8, usually about 10, segments that are randomly chosen from the intracellular pool. Random packaging of 10 segments would result by chance in about 3% of the virions having at least one of each of the different genome segments. So that may seem pretty inefficient, and it is, but so many progeny virions are being produced that it's still a very infectious process. So here we have the entire life cycle being reviewed. The virus buds to cell surface receptors. It's brought in by endocytosis through the movement of hydrogen ions across the membrane, and then ultimately through the M2 pathways. You have low pH, which leads to fusion and uncoating. Then the RNAs are transported into the nucleus. Now remember, this is the first of the RNA viruses that we've talked about, whether positive or negative strand, that are replicating in the nucleus. And that's where viral replication will occur, also transcription of messenger RNA, and ultimately the materials are moved out into the cytoplasm and you have budding from the plasma membrane. People with flu can spread it to others up to about 6 feet away. Most experts think the flu virus spreads mainly by droplets made when people with flu cough, sneeze, or talk. These droplets can land in the noses or mouths of people who are nearby or possibly be inhaled into the lungs. Less often, a person might get flu by touching a surface or object, a fomite, that has flu virus on it, and then touching their own mouth, nose, or possibly their eyes. People with flu are most contagious in the first 3 to 4 days after their illness begins. Most healthy adults may be able to infect others beginning 1 day before symptoms develop and up to 5 to 7 days after becoming sick. Children and some people with weakened immune systems may pass the virus for longer than 7 days. Symptoms can begin about 2 days, but range from about 1 to 4 days after the virus enters the body. That means that you may be able to pass on the flu to someone else before you know you are sick, as well as while you are sick. Some people can be infected with the flu virus but be asymptomatic. During this time, those people may still spread the virus to others. Here you can see some of the pathology that can be caused by infection with the flu virus. The top picture shows the normal trachea of an individual. You can see all the cilia, but 3 days post-infection, you can see desquamation and loss of cilia. But within a few days after infection, this starts to reverse and you can see the cilia are growing back. But the virus itself can have tremendous impact on the respiratory system of an infected person. Every year in the United States, anywhere from 5 to 20% of the population will get the flu. We have a couple 100,000 people generally who end up being hospitalized. We have thousands of people who die from the flu. We're seeing increases in recent years in morbidity and mortality, and we think that that has to do with changes in the population rather than changes in the flu. So, for instance, our population in the United States is getting older. We see a graying of the population, and elderly people are more likely to have severe infections and perhaps die from flu. People with cystic fibrosis live longer now because of various kinds of interventions that have been developed, and those individuals are more sensitive to the flu. We have more and more high-risk neonates being born every year. And we have a greater portion of the population that is immunosuppressed for various reasons, whether it's HIV or various kinds of drug treatments and so on. So it's thought that these factors are playing a role in this increase in morbidity and mortality that we're seeing. But here let's look at 2017-2018. There was, it's estimated that there were 49 million cases, and this resulted in almost a million hospitalizations and almost 80,000 deaths. So, that was a pretty significant flu season. When we look at the symptoms and complications, you're all familiar with the symptoms, but in terms of complications, there are neurological symptoms such as Reye's syndrome, and that's a potentially fatal disease that causes numerous detrimental effects to a lot of organs, especially the brain and the liver, as well as causing hypoglycemia. The exact cause of Reye's syndrome is unknown, but it's been associated with aspirin consumption by children with viral illness. But it does also occur in the absence of aspirin use. The disease causes fatty liver with minimal inflammation and severe encephalopathy. But we have Guillain-Barré syndrome, which is another problem. This is an autoimmune disorder that affects the peripheral nervous system, and what we see is that it can cause usually a transient paralysis in people, usually an ascending paralysis, so it starts with the legs and moves its way upward. But we also have meningitis and encephalitis. People can have pericarditis and myocarditis, so the heart can be affected directly. We have all kinds of respiratory issues, and in pregnancy we see complications that can lead to lower birth weights and so on. And we also have the possibility of myositis, or inflammation of muscle. So there are a lot of complications that can arise from a flu infection. It isn't always a simple disease. In terms of mortality, the causes of mortality are usually bacterial pneumonia or cardiac failure. So these are virus-associated deaths. So remember the impact that the virus had on the linings of the respiratory tract. That makes a person more susceptible to secondary infections such as bacterial pneumonia. We do see that 90% of deaths each year are in people over 65 years of age. Now, if you're wondering if it's the flu or a cold, There are different ways that you can compare these. With the flu, you usually have an abrupt onset. You have a fever, aches and pains, and chills are common, and you typically have a cough, which can be severe, and headache. So, some differences between colds versus flus. Now let's look at how we classify influenza virus. There's 4 different types of influenza virus. There's influenza A, which infects humans, birds, swine, and horses. We see antigenic shift and drift in this group. We'll talk more about that in a minute. And so influenza A is the type of influenza that can cause epidemics and pandemics. So it's the one that we're most concerned about. Influenza B infects only humans. Because it infects only humans, we see antigenic drift but not antigenic shift. So this one can cause epidemics, sometimes serious, but not pandemics. Influenza C is not a serious human pathogen, and influenza D infects mostly cattle. Okay, so now let's talk about how we name influenza viruses. Well, there's the antigenic type. So is it influenza A, is it influenza B or C? So that's first. Then the host of origin. So is it from swine, chickens, and so on? If it's a human origin, then you don't have to write down what the host. You don't give a host of origin designation if it's a human origin virus. You want to give the geographic origin. You want to give it its strain number, the year of isolation, and then for influenza A, we also itemize the hemagglutinin and neuraminidase antigen description in parentheses. So you can see down here, this particular example, the virus type is A, the place it was isolated was Sydney, strain number is 5, the year it was isolated is 97, and this is an H3N2 virus subtype. So hemagglutinin 3 type and neuraminidase 2 type. No animal is given, and so this one would be from human origin. An influenza pandemic is a global outbreak of a new influenza A virus that is very different from current and recently circulating human seasonal influenza A viruses. Pandemics happen when new influenza A viruses emerge, which are able to infect people easily and spread from person to person in an efficient and sustained way. Because the virus is new to humans, very few people will have immunity against the pandemic virus, and a vaccine might not be widely available. The new virus will make a lot of people sick. How sick people get will depend on the characteristics of the virus, whether or not people have had any immunity to the virus, and the health and the age of the person being infected. So we have the difference between seasonal flu, which we always have that, and there may be some immunity built up in the population, and seasonal flu can be of either type A or type B. Then we have pandemic flu. These occur in the human population rarely and unpredictably, but you have widespread transmission because the population lacks immunity, and it tends to affect all age groups, including healthy young adults. This will only be a type A virus. Okay, so let's look at some past pandemics. The 1918 influenza pandemic was the most severe pandemic in recent history. It was caused by an H1N1 virus with genes of avian origin. Although there is not universal consensus regarding where the virus originated. It was spread worldwide during 1918 to 1919. In the United States, it was first identified in military personnel in spring 1918. Remember, World War I was going on at the time. It is estimated that about 500 million people, or 1/3 of the world's population, became infected with this virus. More than 500,000 people died in the United States, and up to 50 million people may have died worldwide. Many people died within the first few days after infection, and others died of secondary complications. Nearly half of those who died were young, healthy adults. Influenza A H1N1 viruses still circulate today after being introduced again into the human population in 1977. In February 1957, a new influenza A H2N2 virus emerged in East Asia, triggering a pandemic. This H2N2 virus was comprised of 3 different genes from an H2N2 virus that originated from an avian influenza virus, including the H2 hemagglutinin and the N2 neuraminidase genes. It was first reported in Singapore in February 1957, then in Hong Kong in April 1957, and then in coastal cities in the United States in the summer of 1957. The estimated number of deaths was 1.1 million worldwide and 116,000 in the United States. The 1968 pandemic was caused by an influenza A H3N2 virus comprised of 2 genes from an avian influenza virus, including a new H3 hemagglutinin, but also contained the N2 neuraminidase from the 1957 H2N2 virus. It was first noted in the United States in September 1968. The estimated number of deaths was 1 million worldwide and about 100,000 in the U.S. Most excess deaths were in people 65 years and older. The H3N2 virus continues to circulate worldwide as the seasonal influenza A virus. Seasonal H3N2 viruses, which are associated with severe illness in older people, undergo regular antigenic drift. So let's look at antigenic drift and shift. So with antigenic drift, we have small random changes in the HA and or the NA. This is through the accumulation of small mutations. Essentially, it occurs because these viruses are passing through partially immune populations, which provides some selective pressure, and you get small random genetic changes in the virus over time. Now, antigenic shift is a much more sudden event. Here you get a major change in the HA, and this most likely occurs through recombination, the reassortment that we see when you have a multi-segmented genome. We've already talked about this process before, and that's what occurs with antigenic shift. And so with all of these different viruses that we see causing pandemics, it's always an influenza A, because remember, that's the type of influenza that not only infects humans, but it also infects a number of different animal species as well. And that contributes to the possibility for antigenic shift. So let's look at some of these types of viruses that have been developing. Well, we have the H1N1 flu. That outbreak began in North America in April 2009 and spread rapidly around the world. By the time the World Health Organization declared a pandemic in June 2009, a total of 74 countries and territories had reported laboratory-confirmed infections. The H1N1 caused high levels of summer infections in the northern hemisphere, which is different from the usual seasonal flu patterns, kind of flipped it. The H1N1 virus has also led to patterns of death and illness not normally seen in seasonal flu infections. Most of the deaths caused by the pandemic flu have occurred amongst younger people, including those who were otherwise healthy. Pregnant women, younger children, and people of any age with certain chronic lung or other medical conditions appeared to be at higher risk of more complicated or severe illness. Many of the severe cases have been due to viral pneumonia, which is harder to treat than bacterial pneumonias that are usually associated with seasonal influenza. So the H1N1 virus has two genes from influenza viruses that normally circulate in pigs in Europe and Asia, along with bird genes and human genes, that way making it a quadruple reassortment virus. So it was an unusual temporal pattern, unusual risk groups, and a quadruple reassortment. So it turned out that the 2009-2010 was an unusual flu season. The percentage of all doctor's visits by patients with influenza-like symptoms dropped from a high of 7.8 in late October to 1.8 in late February, well below the norm for flu season. Flu experts say that this would normally be the time to see the peak of flu cases. Influenza comes and goes in waves, normally running from October through May here in the U.S., but pandemic viruses often move to a different rhythm and can reach their busiest stage at unusual times, like summer or early fall, although the reason for this isn't understood. Flu has peaked in late February or early March in 20 of the last 26 flu seasons. But that swine flu wave started in August and peaked in late October before waning. The H1N flu hasn't disappeared, and another wave could still come, but it was indeed a very strange pattern that we saw with this particular pandemic flu. Avian influenza, or bird flu, is a poultry disease caused by viruses that normally infect birds. This disease is caused by a number of type A influenza viruses. Domestic poultry may be infected with one of two types of avian influenza viruses. The highly pathogenic viruses spread quickly and may kill an entire poultry flock within 48 hours. The low pathogenic viruses cause only mild symptoms in chickens, such as ruffled feathers or a drop in egg production. Although these avian influenza viruses usually do not infect humans, cases have occurred in humans outside the U.S. The disease can be transmitted to humans by contact with infected poultry or contaminated surfaces. Usually, avian influenza virus refers to influenza A viruses found chiefly in birds, but infections with these viruses can occur in humans. The risk from avian influenza is generally low to most people because the viruses do not usually infect humans. However, confirmed cases of human infection from several subtypes of avian influenza have been reported since 1997. Most cases of avian influenza infection in humans have resulted from contact with infected poultry, such as domesticated chickens, ducks, or turkeys, or surfaces contaminated with secretions or excretions from infected birds. The spread of avian influenza viruses from one human to another has been reported very rarely and has been limited, inefficient, and unsustained. H5N1 emerged in 2003 and made it to Western Europe, West Africa, India, and the Middle East by 2006, but began a slow pullback in 2008, leaving only a few pockets of activity outside of Asia, primarily Indonesia and Egypt. Along the way, this virus managed to infect at least 850 people, killing more than half, in 16 countries, although these official numbers are almost certainly an undercount. While some clusters have been reported, H5N1 remains difficult to spread from human to human. Currently, the avian influenza H5N1 virus continues to circulate in poultry in some countries, especially in Asia and Northeast Africa. H5N1, occurring as an epizootic in poultry in some countries, is not expected to diminish significantly in the short term. Thus, sporadic human infections with H5N1 virus resulting from direct or close contact with infected, sick, or dead poultry are expected to continue to occur, and some of these cases are likely to be fatal. Influenza A viruses circulating among poultry have the potential to recombine with human influenza A viruses and become more transmissible among humans. If H5N1 viruses gain the ability for efficient and sustained transmission among humans, an influenza pandemic could result, with potentially high rates of illness and death worldwide. Therefore, the H5N1 epizootic continues to pose an important health threat. The last human case was reported from China with an onset of December 2015. But the U.S. is continuing to stockpile the highly pathogenic H5N1 vaccine. It's not currently available for use, but it could be used if the virus begins transmitting easily and efficiently from person to person. So it's in the stockpile as a potential. Despite a 10-year head start, in 4 short years, H7N9 has overtaken H5N1's lead in reported human infections. Still confined to mainland China, this low pathogenic virus in birds has proven quite deadly in humans, exacting a 40% mortality rate among known cases. Tracking and eradicating H7N9 in wild birds and poultry is extremely difficult, as this virus, unlike H5N1 and many other avian viruses, does not sicken or kill the birds. Often the first hint of local poultry infection comes when humans start to fall ill. Like with H5N1, the true number of infections is unknown, as only the sickest of the sick are ever hospitalized and tested. The H7N9 virus had not previously been seen in either animals or people until it was found in March 2013 in China. However, since then, infections in both humans and birds have been observed. The disease is of concern because most patients have become severely ill. Most of the cases of human infection with this virus have reported recent exposure to live poultry or potentially contaminated environments, especially markets where the birds have been sold. Fortunately, this virus does not appear to transmit easily from person to person, and sustained human-to-human transmission has not been reported. But this virus, like the others, is the result of multiple reassortment events involving multiple hosts, which is a hallmark of influenza A viruses. In 2017, hundreds of outbreaks in domestic and wild birds in 30 European countries were reported, the largest epidemic by a highly pathogenic avian influenza virus in the continent. While some similarities with previous highly pathogenic avian influenza virus epidemics were observed. For example, in the pattern of emergence, significant differences were also noted, in particular, the size and extent of the epidemic. After an absence of nearly 3 years, the virus has re-emerged in 2020 in West Central Europe and has thus far been detected in 5 European nations since the start of the year. The latest nation to be struck is the Czech Republic. While currently considered a low-risk virus for human health, it can be devastating to poultry, and there are always concerns that the virus could evolve over time, posing a bigger threat to humans. Dog flu is a contagious respiratory disease in dogs caused by a specific type A influenza virus referred to as a canine influenza virus. This is a disease of dogs, not of humans. It is an influenza A H3N8 influenza virus that was originally an equine or horse influenza virus. This virus has spread to dogs and can now spread easily between dogs. The H3N8 equine influenza virus has been known to exist in horses for more than 40 years. In 2004, however, cases of an unknown respiratory illness in dogs, initially greyhounds, were reported. An investigation showed that this respiratory illness was caused by the equine influenza A H3N8 virus. Scientists believe that this virus jumped species from horses to dogs and has now adapted to cause illness in dogs and spread efficiently among dogs. This is now considered a new dog-specific lineage of H3N8. In September of 2005, the virus was identified by experts as a newly emerging pathogen in the dog population. The symptoms of this illness in dogs are cough, runny nose, and fever. However, a small proportion of dogs can develop severe disease. The number of dogs infected with this disease that die, fortunately, is very small. Some dogs have asymptomatic infections, while some will have more severe infections. Severe illness is characterized by the onset of pneumonia. Although this is a relatively new cause of disease in dogs, and nearly all dogs are susceptible to infection, about 80% of dogs will only have a mild form of the disease. Canine influenza virus can be spread by direct contact with respiratory secretions from infected dogs, by contact with contaminated objects, and by people moving between infected and uninfected dogs. Therefore, dog owners whose dogs are coughing or showing other signs of respiratory disease should not participate in activities or bring their dogs to facilities where other dogs can be exposed to the virus. Clothing, equipment, surfaces, and hands should be cleaned and disinfected after exposure to dogs showing signs of respiratory disease. To date, there's no evidence of transmission of canine influenza virus from dogs to people, and there has not been a single reported case of human infection with the canine influenza virus. So let's go back to this question. I talked about this a little bit when we discussed the different types of influenza. But remember, we only have pandemics caused by influenza A, not influenza B. And so far, no shifts have ever been reported amongst influenza B. And there's no known animal reservoir for influenza B. Influenza B viruses appear to be strictly human viruses. So what we have in terms of the evolution of new strains is through the process of antigenic drift, which is a slow process, as opposed to shift, which can cause very quick, major changes in a virus. In terms of preventative steps, if you're sick with a flu-like illness, you should stay home for at least 24 hours after your fever is gone, except to get medical care or for other necessities. You want to avoid close contact with people and so on. You're all familiar with these sorts of things. But at the bottom there, vaccination. That's an important step. There are a number of different kinds of vaccines that you can now choose from. Over the past few years, several different methods have been developed. For older people, we have a high-dose vaccine that contains 4 times the antigen as the standard inactivated influenza vaccine. There's another type of shot targeted for the elderly population that has an adjuvant added to help create a stronger immune response to the vaccination. For these shots, as well as the standard flu shot, this year they're going to be quadrivalent, which means that they will be immunizing against 4 different strains of virus. Another kind of vaccine that's available is one that's grown up in cell culture. A potential advantage of cell culture technology is that it might permit faster startup of the vaccine manufacturing process in the event of a pandemic. The cells used to manufacture the vaccine are kept frozen and banked. Cell banking assures an adequate supply of cells is readily available for vaccine production. Using cell culture has the advantage that it removes the dependence on an egg supply to produce it. In the event of a pandemic caused by an avian flu, it's possible that the egg supply could be affected by bird mortality. Also, viruses grown in eggs often develop egg adaptations that can reduce their effectiveness. The viruses used to make cell-based vaccines may be more similar to circulating wild flu viruses than the viruses used to make egg-based vaccines. The recombinant vaccine uses an HA gene from a naturally occurring or wild-type recombinant vaccine virus. This HA gene is then combined with portions of another virus that grows well in insect cells. This recombinant vaccine virus is then mixed with insect cells and allowed to replicate. The flu HA protein is then harvested from these cells and purified. The purified protein is packaged while waiting for FDA testing and approval to release lots. Currently, the recombinant flu vaccine is the only 100% egg-free vaccine in the U.S. market. And then we have the live attenuated influenza vaccine, and this is only available as a nasal spray. So there are many different kinds of choices now that people can make. So one question is, how are the viruses selected to go into the flu vaccine each year? The flu viruses selected for inclusion in the seasonal flu vaccines are updated each year based on information about which influenza viruses are being found, how they are spreading, and how well the previous season's vaccine viruses might protect against any that are being newly identified. Currently, more than 100 national influenza centers in more than 100 countries conduct year-round surveillance for influenza viruses and disease activity. These laboratories then send influenza viruses for additional analysis to 5 WHO collaborating centers for reference and research on influenza. Vaccine viruses are chosen to maximize the likelihood that the influenza vaccine will protect against the viruses most likely to spread and cause illness among people during the upcoming flu season. Scientists use the hemagglutination inhibition test. Remember that one? There was a reason why I made you learn about that. But they use that test to assess the antigenic similarity between influenza viruses. This test is particularly useful for helping to select the vaccine viruses used in the seasonal flu vaccine. HI results can tell us whether antibodies developed against vaccination with one virus are antigenically similar enough to another circulating influenza virus to produce an immune response against that circulating virus. Scientists also use the HI test to compare antigenic changes in currently circulating influenza viruses with influenza viruses that have circulated in the past. CDC conducts studies each year to determine how well the flu vaccine protects against flu. While vaccine effectiveness can vary, recent studies show that flu vaccination reduces the risk of flu illness by between 40 and 60% among the overall population during seasons when most circulating flu viruses are well matched to the flu vaccine. In general, current flu vaccines tend to work better against influenza B and influenza A H1N1 viruses and offer lower protection against influenza A H3N2 viruses. How well the flu vaccine works, or its ability to prevent flu illness, can vary from season to season. The vaccine's effectiveness can also vary according to who's being vaccinated. At least two factors play an important role determining the likelihood that the flu vaccine will protect a person from illness. First, the characteristics of the person being vaccinated, such as their age and health, and second, the similarity or match between the flu viruses and the flu vaccine that is designed to protect the community against the viruses spreading in the community. In terms of priority groups, children are in the priority group. They need to be at least 6 months old, though, to receive the vaccine. Pregnant women, people over 50, people with chronic conditions. Of course, people who live in nursing homes are high priority, and people who live with or care for those at high risk for complications, so healthcare workers and people who have household contacts with people who are at high risk and so on. There are several prescription medications that can be used to treat flu illness. CDC recommends prompt treatment for people who have flu infection or suspected flu infection and are at high risk of serious flu complications, such as people with asthma, diabetes, or heart disease. Antiviral drugs are prescription medicines. Some are pills, there's a liquid, and one is inhaled as a powder, or another as an intravenous solution. And they all fight against the flu viruses in your body. None of these antiviral drugs are sold over the counter. You can only get them if you have a prescription from a healthcare provider. And of course, antiviral drugs are different from antibiotics, which fight against bacterial infections. Now we have 3 that are neuraminidase inhibitors: Tamiflu, Relenza, and Rapivab. These are chemically related drugs, and they work against both influenza A and B viruses. They block the active site of the influenza viral enzyme neuraminidase, which is common to both influenza A and B viruses. This effect results in viral aggregation at the host cell surface and reduces the number of viruses released from the infected cell. When used within 48 hours of illness onset, these drugs decrease shedding and reduce the duration of influenza symptoms by approximately 1 day compared with the placebo. They also reduce complications. They can be used for chemoprophylaxis, but are not a substitute for a vaccine. They can be used as a component of an outbreak control program, for instance, in a chronic care facility. The U.S. maintains a stockpile of Tamiflu, for instance, in case of a pandemic. Balaxavir is another antiviral drug, and it works as an enzyme inhibitor targeting the influenza virus's cap-dependent endonuclease activity. that's used in cap snatching by the virus polymerase complex, a process that's essential to its life cycle. So this is an example where I told you in the beginning of the course you had to understand how a virus replicates in order to come up with treatments. And influenza provides a wonderful example. We have all these different drugs that have been developed to inhibit the replication of influenza, but they all required the understanding of the molecular replication of the virus in order to identify these drugs. Isolation and quarantine are two common public health strategies that are designed to protect the public by preventing exposure to infected or potentially infected persons. Under certain conditions, isolation and quarantine can be used in the case of influenza. The recent spread of highly pathogenic strains of avian influenza has highlighted the threat posed by pandemic influenza. In the early phases of a pandemic, the only treatment available would be the neuraminidase inhibitors, which many countries are considering to stockpile for pandemic use. As I mentioned, Tamiflu is stockpiled by the U.S. It's estimated that stockpiles that cover 20 to 25% of the population would be sufficient to treat most of the clinical cases and could lead to 50 to 70% reductions in hospitalizations. Substantial reductions in hospitalization could be achieved with smaller antiviral stockpiles if drugs are reserved for persons at high risk. And again, we need to be concerned with reducing hospitalizations for the same reasons that we're concerned about reducing hospitalizations as a result of COVID. If the hospitals become overwhelmed, then you not only can no longer take care of the patients who are suffering from the flu, but it also impacts care for patients who are dealing with other kinds of illnesses and injuries. There are 5 categories of influenza surveillance that occurs in the U.S. The first is virological surveillance. There are laboratories located around the world and throughout the United States that participate in virologic surveillance for influenza. A key part of this surveillance is the surveillance that's done for novel influenza A viruses. In 2007, human infection with a novel influenza A virus became a nationally notifiable condition. Novel influenza A infections include all human infections with influenza A viruses that are different from currently circulating human seasonal influenza H1 and H3 viruses. Rapid reporting of human infections with novel influenza A viruses will facilitate prompt detection and characterization of the viruses and accelerate the implementation of an effective public health response. Then we have outpatient illness surveillance. Information on patient visits to healthcare providers for influenza-like illness is collected through the U.S. Outpatient Influenza-like Illness Surveillance Network, or ILINET. ILINET consists of more than 2,900 outpatient healthcare providers in all 50 states, Puerto Rico, the District of Columbia, and the U.S. Virgin Islands, reporting more than 30 million patient visits each year. Mortality surveillance is done. Rapid tracking of influenza-associated deaths is a major way to keep track of what's going on. Hospitalization surveillance is done. Laboratory-confirmed influenza-associated hospitalization in children and adults are monitored through the Influenza Hospitalization Surveillance Network, or FluSurvNet. And then we have the summary of geographic spread of influenza. State health departments report the estimated level of geographic spread of influenza activity in their states each week through the state and territorial epidemiologist reports. States report geographic spread of influenza activity as no activity, sporadic, local, regional, or widespread. And you can see that in that figure up on the top right here. And then at the bottom, you can see the kind of information that's reported to ILINET. Okay, then to finish up on this lecture, we've discussed 3 different virus families: Bunyaviridi, Arenaviridi, and Orthomyxoviridi. And for obvious reasons, I put them together. They have significant shared properties, including the fact that they are multipartite, negative sense, single-stranded RNA genome viruses. They all have an envelope, and they all use cap snatching to transcribe their messenger RNA. But there's a big difference. The orthomyxoviruses replicate in the nucleus. This is the first RNA virus that we've talked about that does this.